We have one more team left today, Team IO Target, and they're coming today from the Birnbaum, Irvin, White, and Wittrup labs. So feel free to take it away.
All right. So this is Michael, and I'm Forrest. Sorry, I'm losing my voice, but it's getting better, believe it or not. It was worse yesterday. Anyway, so we're here to present IO Target, and we basically have a revolutionary technology that will shrink solid tumors, prevent metastases, improve hair growth, at least for eyebrow hair, and improve your social life. So I think at least one of these is true. So the technology is really founded on immunotherapy, which you've heard a lot about over the last 5 talks, some more than others. And the immunotherapy that we're particularly interested in is technology that's been developed by the Irvine and Wittrup labs over the last couple of years. And it's sort of a multi-angled approach at immunotherapy, where they're treating tumors now with these lymph node targeting peptide vaccines that you heard about from Naveen and Jaydeep. And basically these vaccines, if you know the antigen, you can stimulate the lymph node to vaccinate against that antigen. These checkpoint inhibitors, basically, that activate T cells that have been suppressed in the immune environment. Extended half-life IL-2 that increases the activity of adaptive immune cells as well as the innate immune cells. And then these anti-tumor antibodies that basically coat the tumor and then recruit adaptive immune cells to the tumor. And the amazing thing about this approach that they've developed is that if you take mice and infect them or give them a B16 F10 melanoma xenograft, and then you wait 8 days and let the xenograft establish, you can then treat with this multi-agent immunotherapy and treat once per week, basically. And the amazing thing that you can see here is that mice that have been given this treatment, the tumor basically shrinks and then goes away. And so they're basically able to cure these mice with this B16 F10 melanoma. Go ahead. Michael's going to interrupt me here and take over. These junior faculty, I don't know what they're thinking. But anyway.
Hi. So, in an attempt to alienate half the room and get fired, what I have to point out is that this is super great news if you happen to be a mouse xenografted with a tumor line. For the regular mice of you out there, let alone the people, this isn't going to hold. And so we have really a great collection of technologies, both the ones that are up here, some of the ones that some of our competitors talked about, and just in a worldwide push for immuno-oncology that are ready, and it's basically a lot of weaponry without any target to know where to go. And the current standard of how to deal with this is something like this. So the way that people are trying to address this is through neoantigens. And so those are antigens which were mutated in the course of just the general genomic instability of the tumor. So you have antigens which are made by coding differences in proteins all throughout the tumor. You sequence to get them, predict what would bind onto an MHC, which is the target for a T cell. And then you target those antigens. And this is an idea which is very intuitively attractive. And it's an idea that's saving some number of lives, or at least preventing worsening of some cancers. And it's ideas that have been publicized by both or being developed by both very large companies as well as many, many startups. So this is, at the very least, not a terrible idea. This is probably, I just alienated the other half of the room. This is probably even a good idea, but it has some real significant limitations. So one is tumor antigen heterogeneity. So if you have a mutation that was made somewhere in the progression of the tumor that wasn't there at the start, you have a founder effect. So if you develop something that's going to expertly get rid of green cells, you're still left with your blue tumor cells, nothing to do with those. Antigen downregulation, because a lot of these mutations which are found aren't necessarily things that are important for the development or for the proliferation of the tumor. So that provides a selective pressure, much like when you're infected with a virus, for these cells just to one way or the other downregulate that type of signal. And then the other one, which really kind of annoys me, is variability between individuals. So I could put through a lot of effort sequencing my green silhouetted woman here and find her a cure, and it's not going to get me any closer to blue silhouetted man. And I support personalized medicine and I support treating cancer, but I really do find it irritating that that's the best we think that we can do in a lot of these cases, that a lot of these approaches really do rely on going one by one rather than larger populations. And I didn't have a great answer for this, but some of that Koch Institute magic, which I'm finding out about, is that my neighbor next door did. So I'll let him talk more.
All right, thanks. So our approach to this is to take a little bit more intelligent approach, I'll say, without patting ourselves on the back too much, which is basically to go after some of the post-translationally modified peptides that are going to be naturally presented by the immune system. So where are these post-translationally modified peptides coming from? They're from the activated signaling networks that are known to be necessary to drive tumor proliferation, tumor migration, tumor invasion. All those classic tumor phenotypes are driven by activated kinases, activated acetyltransferases, glycosylases. A variety of different enzymes lead to different protein post-translational modifications, and these then drive these phenotypes. These PTM-modified proteins are obviously subject to proteasomal degradation, just like all other proteins, and they get cut up into peptides. And some of those peptides contain the post-translational modification site. And just like any other proteasomal degraded peptides, some of them can get loaded onto MHC class I molecules, trafficked to the surface of the cell, and displayed to the immune system. And previously, what we've done is actually, when I was a postdoc in University of Virginia, we identified multiple of these phosphorylated MHC antigens that are presented on the cell surface. And so basically, what we aim to do here is to take tumors, lyse the tumors, immunoprecipitate out these MHC class I peptides, peptide complexes, go in, affinity enrich for the PTM modified peptides, and then use LC-MS/MS to identify those peptides, and most importantly, quantify those peptides for their level of expression on tumors versus normal cells. And through doing this, we can identify these novel tumor antigens that are presented on the tumors. One of the critical things that Michael mentioned that I need to stress here is that in immune evasion, if we just have a passenger mutation, it's really easy for the tumor to mutate away from that passenger mutation. But it's actually difficult for the tumor to mutate away from these activated signaling networks, because these are the things that are critical for driving the tumor to proliferate, to migrate, and do everything else. So even if we lose a single EGFR phosphorylation site, for instance, the tumor basically then turns that pathway back on and reinvigorates that same EGFR phosphorylation in order to survive. So Michael's going to tell you what we can do with these PTM antigens once we have them.
So once Forrest works his magic, and you might ask, by the way, why nobody has tried this before, and it's partially because you could have the world's brightest lamppost, and it's not going to help you find things that are not under, right? Neoantigens and things like that, we know how to find them. So we think we have another way. We'll find them, and then we're going to be able to take them, verify that they actually do bind to MHCs, and then pick out the different T cells which are able to recognize this PTM MHC, as we're calling it here. Take out those T cells and just first functionally characterize them. Understand, are these T cells present in a tumor or lymph nodes or in the periphery? Are they active in a cancer model? What is their phenotype? And we know how to do this. And more than that, we could take the interesting T cells that are overrepresented or have a good activation phenotype in the presence of these molecules, and then take their T cell receptors, express them, and then use those to find other peptide MHC binders using a yeast display technology, which we use in my lab, in order to find potentially optimized peptides which are able to induce an even stronger immune response than potentially the PTM peptide MHC that was originally there. And we know how to do this too. And one of the benefits of this is that there's nothing that says that this is the be-all, end-all antigen for the T cells we want. But there's nothing that says we have to use that to activate the T cells anyway. So long as we get the right T cells activated, we could find things through just experimentally, things that are potentially even 50-fold more potent at activating a given T cell of interest than the natural antigen itself. And so we can characterize the ones that we want, assay whether they're better or not, and then use them to go back into some of the real great technologies that the Irvin and Wittrup groups have created in order to look at this. And more than that, I ran out of room on one of my 5 slides, is that we can also go back and reverse this too. So let's say that you want to make a really great chimeric antigen receptor, so a great CAR T cell, right? These are great targets because these are potentially proteins which are phosphorylated that wouldn't otherwise be phosphorylated. So we can take a T cell receptor and then make it super high affinity and good specificity for these guys too to create your own synthetic killing machines for cells that express the PTM-MHC. Once we have these molecules, we can then go back, and the model we're showing you here today would be a breast cancer cell model, and we could ask questions such as, if you take the immunotherapy, which Forrest told you about at the very beginning, and either try it directly on primary tumors or remove primary tumors or otherwise reduce them through chemotherapy or through surgery, are you able to prevent recurrence? So are you able to then vaccinate to make sure that you won't have either recurrence at the primary site of tumor or metastasis later on or anything like that? Or to steal just like 1% of the team before us, I'm really bad at pronouncing exotic names. That one. To steal a little bit of their thunder, are you able to go beyond preventing recurrence and use these potentially molecules which otherwise wouldn't be phosphorylated or activated as a means for a prophylactic vaccine? So with that, you've been sitting an awful long time, and we thank you for that. We thank the people who are judging, who are hosting, the people watching upstairs in the building. And we're happy to take any questions.
That's awesome.
So I'll just throw a couple of questions, and then we'll have time for others as well. I guess as I was listening to this, I was thinking, why aren't these post-translationally modified proteins just neoantigens? Because they just didn't know they were neoantigens. But effectively, I understand that you're ascribing some, kind of more, let's say, immutability to them based on the function. But for example, will neoantigen discovery people find the same post-translational modifier? I know they won't find them by sequencing,
right.
But so that's one general area. The second quick thing it would be good to comment on is, how much specificity has been shown in terms of tumors being associated with these PTMs that really separate them, because otherwise these things would be used for diagnostics. Absolutely. And there's a signal-to-noise issue there. And then the third very quick thing to comment on is, in your report, you kind of have 3 aims, but the third aim looks like it needs a lot of money. I want to know what's the chance that, because that's kind of the proof that you want to get, what's the chance you get halfway across the river and then kind of are stranded there, in the sense that you show there's these exciting PTMs, and you show that they have T cells that you can even make better peptides against, and then you can't really show that they do any better than what people are doing today.
Yeah, all right, so I'll try and answer those in order if I remember them all. So the first one, these PTM peptides are exactly neoantigens. They're just not mutant neoantigens. And why do we think they're going to be sort of immutable and have a difficult time with immune evasion is because, for instance, in that EGFR pathway that was up on the screen there, EGFR activates the MEK-ERK pathway. And when you basically target EGFR and try and shut it down, the cell responds by basically upregulating other aspects of that same pathway to drive that same pathway. So if you have a phosphoantigen from downstream in that pathway, it's likely that it's going to be turned on and needed to be turned on in order for the cell to survive. If it loses that activation of that pathway downstream, then the cell's no longer proliferating. So for the cancer to continue proliferating and basically to have a survival advantage, it needs to have that pathway on. I forget the second one.
How specific have PTMs been? You've been studying
Oh
them
yeah,
for
how
20 years.
specific. Yeah, so it's interesting. There's been a few papers. In fact, I think there's been 5 papers, maybe, all out of the same lab, the lab I did my postdoc in at University of Virginia, that have identified some of these phosphorylated antigens. And they've shown a little bit of overlap between different cell lines, but they haven't actually gone to tumors yet, and largely because they don't have the sensitivity to do that. And what we've done in my lab over the last 10 years is basically really radically improve the sensitivity of the mass spec approach that we've been taking. So now we can take biopsies and look at some of these signaling networks from small biopsies. Using that same technology, we could take some of these biopsies. I believe where we're going with this is to take these biopsies and be able to identify the phosphorylated or acetylated or other PTM modified antigens.
And the third question was, your third aim says that it's to show prevention of cancer recurrence in some
Exactly.
model.
So basically, as soon as we win this, as soon as the money comes in, we're basically ready to go to basically start identifying some of these antigens. And what Daryl and Dane's lab have been waiting for are basically antigens from the surface of these tumor cells. So they're ready to go with the tumors, we're ready to go with the technology, and we're basically aiming to do AIM 3, even as Michael's working on AIM 2. So it's really not stepwise. It's sort of, once we identify some of these antigens, we can start cranking.
Other questions from the group? You can answer questions from the previous time, too.
So I have sort of a philosophical question, right? So the strategy here is amp up the immunogenicity, right? That's sort of how I look at this. You got the cancer, you got the immune system. The cancer's not being immunogenic enough. This is a way to amp up the immunogenicity.
It's a way to target the immune system specifically to the cancer.
So you think that's the problem? Or because the reason, it's interesting that the reason current immunotherapy seems to work is not because the cancer is not immunogenic enough, it's because something else is shutting down or is attenuating the response. So is the problem making the cancer more immunogenic, or is the problem something else?
Do you want to take this?
Yeah, I'll take this one. I think that it's both, but I think that something that a lot of the approaches which are trying to take the brake off of the immune response to try to defeat some of the mechanisms that cancer uses to either dampen or evade an immune response. The problem there is that they're quite antigen non-specific, basically all of them. And so you're left with potentially treating the tumor, but also having, depending on the treatment, really significant morbidities and mortalities of just, essentially autoimmune phenotypes. And we accept that because we think that autoimmunity is better than dying of cancer, and I agree. But we think we could do better than that too, because we think that if you couple it and basically find the right combination, you can remove the brake, but then also really give a kick in the pants to the right set of immune cells. And what we think is missing for that is targets such as the ones which we're trying to get. So we think that it is both, but we think that we could do a better job than just simply, some of the checkpoint blockade, which has made incredible progress, along with some other things which are being developed in terms of, turning off immunosuppressive signals in the tumor microenvironment or something like that. But we think that even if that is an important mechanism, we add to that as well.
So just one more comment, because I realized I didn't address your diagnostic question. And if we find, for instance, that because many of the same pathways that we would be interested that might appear as phosphorylated peptides, the MEK-ERK pathway, for instance, the PI3-AKT pathway, a variety of other pathways that are known to be activated in these tumor cells and might therefore appear on the surface of the cell. If we find some of those that are truly across many different cancers, then we actually have a way of basically not having patient-specific precision medicine, but instead having a generalized therapy that could be used for many different patients and many different cancer types, and therefore for diagnostic purposes as well. And also, this gives us basically tumor-specific addresses. So in thinking about trying to deliver particular nanoparticles or whatever to the tumor, if you had a tumor-specific address on the surface of the tumor, this provides you with another way of basically using this information that we're generating.
Thank you so much.
All right.
So thank you to all of the teams that presented this morning. This has been really exciting to listen to. As a science writer, I feel like my biggest challenge on a daily basis is understanding the ideas that scientists have and then what potential impact they could have, and then also what limitations they have. So this has been really wonderful to listen to. So now it's up to the judges to deliberate. So they're going to be excused to chat privately, and while they're doing that, there'll be a video playing on to see what else is going on at the Koch Institute. Or stretch, whatever you.